Welcome back to our third installment of our Chapter 3 Genetics lectures. This one is all about meiosis. So this is going to be a general overview of meiosis, non-disjunction, polyploidy, all of that fun stuff. So just to kind of go over the general definition of meiosis and why we should care about it is meiosis is where sex cells are made in reproductive organs. It's a reduction division of a diploid cell into 4 unique haploid nuclei, 4 haploid daughter cells. This is what becomes your egg and sperm cells. During your first bit of meiosis, when you're in interphase, DNA is replicated. When DNA is replicated, you get 2 genetically identical copies of your DNA. And these 2 copies are called sister chromatids. They're held together by a centromere, and they're homologous to each other. Now, I'm not going to go through each individual step. You've learned this before. This is just a general overview. So you start with interphase, where DNA is replicated, and then you go through your first division, which is prophase, metaphase, anaphase, and telophase. And then you split those cells, right? You go from 1 cell to 2 cells, and then you go through the whole thing again. And then you go through prophase 2, metaphase 2, anaphase 2, and then telophase 2. And then when you go through cytokinesis your second time, you end up with your 4 cells. In prophase 1, so that first step really in meiosis after interphase, homologous chromosomes can undergo what's called crossing over. So homologous chromosomes pair up to form a bivalent. That's just when you have 2 homologous chromosomes lined up together. Those homologous chromosomes are held together at a point called a chiasmata, and then crossing over of genetic material can happen. So you cross over material between non-sister chromatids, and that's how you get a little bit of genetic variation. And you can see that here you have now a red leg on the blue chromosome and a blue leg on the red chromosome. From here, another version or another way you can get genetic variation is through what's called random assortment. So during metaphase 1, homologous chromosomes line up at the equator of the cell. Now, whether the mom version or the dad version ends up on the left or the right is totally random. And because this is random, you get a little bit more of genetic variation. And you can see that in the 2 possibilities. So you could have, let's say dad is blue and mom is red. You could have the dad on the left for both chromosomes and mom on the right. And then you get all of mom or all of dad. Or you could have a little bit of a mix-up, and then you get a little bit more of a combination between mom and dad in your haploid cells. So, general overview of mitosis versus meiosis. I'm not going to go through this whole thing, but you should pause and make sure that you know all of this. A great way to remember all of this is this acronym at the beginning. All of those bolded terms spell out disco pug. And you can kind of use that to remind yourself of all of the main differences between mitosis and meiosis. Now, mutations. We talked a little bit about mutations already, but they really kind of come down to 2 classifications, somatic or germline. So somatic means that it's happening in a single body cell, and germline means that it's happening in your gametes. Well, if you have a mutation that happens in a single body cell, that's it. It just happens in that body cell, but it can't be inherited and it can't be passed on to offspring. But if you have a mutation that happens in the germline, that means it happens in the gametes. And if it's happening in the gametes, it can absolutely be passed on to your offspring. So the life cycle for sexually reproducing organisms, most of them are diploid, and then in order to reproduce, they need to make gametes that are haploid. Once you go through and you make those haploid gametes, the fertilization of 2 haploid gametes result in a diploid zygote, and that diploid zygote goes on to become your offspring. Just to recap what we were talking about, there are 3 main sources of genetic variation when dealing with sexual reproduction. You have crossing over, random assortment, and then random fusion of gametes from different parents. So that is literally the definition of reproducing with someone that you are not related to. So different parents means that you get gametes from different people. Now let's start talking about some issues that arise if any mistakes happen when you're going through meiosis. So when chromosomes fail to separate correctly, it's called non-disjunction. And what happens is that gametes end up with one extra or one missing chromosome. And you can see that here, right? Non-disjunction meiosis 1, you end up with an extra chromosome over here and one less over here. So you have a +1 and a -1. If this happens in meiosis 2, then the same thing can happen, except you have a higher chance of having some what would be considered normal gametes, and then you have one gamete with an extra chromosome and one with one less. So if it happens in meiosis 1, all of the gametes will have either an extra or one less chromosome. So some conditions that you may have heard of that come from non-disjunction are Patau syndrome, which is trisomy 13, meaning you have 3 chromosomes for 13, Edwards syndrome, which is trisomy 18, Down syndrome, trisomy 21, Klinefelter and Turner syndrome both deal with your sex chromosomes. Klinefelter is when you get an extra X, and Turner syndrome is when you have one less X and you end up with only one X total. Now, because we're talking about sexual reproduction, it is of course important to talk about the age of parents and how that can impact non-disjunction happening in offspring. So there's a strong correlation between your maternal age and the occurrence of non-disjunction, meaning that the older the mother is, the more likely an occurrence of non-disjunction is going to happen. So in this, it's basically saying that abnormalities in the chromosomes can increase significantly after the mother is of age 30. Now, if you're looking at all these charts, you'll notice that, yeah, 30 is like sort of where it starts to happen. 30 is like right here, you start to see an increase. Over here, 30 is like in the middle here, you start to see an increase. But it's not like detrimental. But as you get older, you do see an increase. You do see this kind of exponential growth of occurrences of non-disjunction. So there is a correlation between age and occurrence. For karyotyping, we talked about this last time, but this is how you can test fetuses for the gender and for chromosomal abnormalities. There are 2 main ways. There's chorionic villi sampling, which removes placental tissue. And you can do this pretty early on. It's about 11 weeks. And then amniocentesis, which removes amniotic fluid, which contains fetal cells. This is done a little bit later, around 16 weeks, and you can see the difference between these two. So the villi sampling happens when the fetus is not really fully developed. You can't totally see the features that you would expect to see in a fetus, whereas the amniocentesis, it's a little bit later, so the fetus is a little bit more developed at that point. And then polyploidy is our last bit that we're going to talk about. We have discussed this a little bit before, but polyploidy is this idea where an organism has more than 2 complete sets of chromosomes in all of their cells. So haploid is 1 set of chromosomes, diploid is 2 sets, polyploidy is more than 2 complete sets. This is pretty common in plants because they don't have separate sexes and they can self-pollinate. So polyploidy happens way more frequently. Whereas in animals, it's pretty rare. And I mean like pretty rare, because there are some serious consequences for us for having extra allele copies of every gene. Even just having like 1 extra copy of 1 chromosome can lead to different disorders. So having an extra copy of every gene would be pretty detrimental. So it doesn't really happen in animals. All right, that's all for today. I'll see you next time.